Thank you. There we go.
You need to say any first words too.
Okay, looks like it's recording. Tell me when to start.
Whenever you're ready, go for it.
Okay, so tomorrow. Okay, awesome. We're all here. All right. So today I have the honor of introducing our speaker for the day, Madyson Miller, shown here bragging about robbing a ship full of wing conch with her band of parrots. She she got her bachelor's degree in marine biology from the University of West Florida, and then spent about 6 months working on salt marsh birds, which trained her very well for walking around the mud in California when we went this past summer, and sifting through algae and amphipods. Now, Madyson is a very avid Pokemon hunter, and she's applied the same skills to getting fellowship. So she got a Folk Education Foundation fellowship. She has an NSF STEM grant. She got our Ocean International Policies Summit fellowship that took her to Hawaii. She's been very active in outreach in marine sciences, and that is a passion that has permeated through everything she's done. She's worked for OCOVI as an outreach coordinator. She's an ambassador for Women in Ocean Sciences, and she's very involved in the United Nations Ocean Decade, where she's been a U.S. Youth Advisor as part of a larger international effort. And all these efforts have paid off. And at the end of this year or the beginning of next year, she will be joining the ranks of the well-paid, going to Washington on a very prestigious NAUS Marine Policy Fellowship. She will be in the Coral Conservation Division of NOAA. So she's going to be helping shape policy that is going to affect all of us and everywhere else there's coral reefs for the next several nations. Now, earning the trust of a free-ranging Madyson is not easy. And what I'm showing you here is this award-winning footage from these are University of Colorado scientists making first contact with a free-ranging Madyson Miller in the Bodega Bay Marine Sanctuary. You have to be careful about how you approach these organisms. And eventually you can earn their trust. If this doesn't work, I found out that burritos and vegetarian burgers work very well. Now, Madyson is going to be talking about part of what she's accumulated in terms of data. She has so much data she won't be able to show it all, but she didn't feel that she was ready. When I told her that she was ready to defend, I was like, and she was like, and then I was like, look, and she was like, she finally gave me a manuscript, and I was like, and she was like, and she finally explained things, and then I was like, and she was like, so the goal after all of today is that at the end of today, she will be like,
Oh my God.
it is incredibly, I feel incredibly proud to be introducing the only student in marine sciences who's been DNA barcoded, and my collaborator and future colleague for many years, I hope, Madyson Miller. Take it away.
Follow. Okay, I got it. Can everybody on Zoom see that?
Yes, looks good.
Yes, we see it fine.
Yes. Awesome. Well, thank you, Edwin, for that introduction. Definitely calmed my nerves a little. That was hilarious, and we definitely need to show the people from Bodega that, because that was just so silly. Thank you for taking me on that trip as well. Thank you to my committee members, Dr. Turner and Dr. Stanford, for helping me get to this point that I'm at. And also thank you all of my friends and family. I think I have some family on Zoom for coming today. Okay, let's get right into it. Try to get rid of. Okay, I don't know if it's going to go away, but let's get right into this. So seagrasses are marine flowering plants, and they can be found worldwide, including right here on St. Thomas in our own backyards. Seagrasses are different from seaweeds. Seagrasses are different from seaweeds in that they have different reproductive abilities and different structures. In fact, they're more like terrestrial plants because they have really dense root systems, stems, and leaves. These qualities allow them to build really beautiful seagrass meadows like the one shown in this picture. And these seagrass meadows are important because they provide numerous ecosystem services. For example, they can store carbon, not garbage. They can store carbon, which will help to mitigate climate change. And they can also store sediment, and this helps prevent soil erosion, which in turn helps protect our coastlines. Seagrasses are primary producers, which means they make their own food from sunlight. This promotes primary production in the ecosystem. Now, all of that's great, but something else that they do that's also great is that they are an important food source and nursery ground for lots of different herbivores, including the ones that we like to eat, so juvenile fish, the really cute itty bitty invertebrates, and also the really cute megafauna like sea turtles and the endangered manatees or the chubby mermaids. Unfortunately, seagrasses are in decline worldwide. This is primarily due to the indirect and direct impacts of man-made stressors, also known as anthropogenic stressors. A few of those include climate change and habitat degradation. Okay, spoiler alert. In the US Virgin Islands, we have three really common marine native seagrasses: Thalassia, Serengodium, and Halophila. We also have two species of Halophila. The native species is Halophila decipiens, and the non-native species and star of my thesis, drumroll please, is Halophila stipulaceae. Halophila stipulaceae is native to the Red Sea, Persian Gulf, and Indian Ocean. So you're probably wondering, how the heck did it get to the Caribbean? Well, it most likely came by the dumping of ballast water from large shipping vessels. It was initially reported in 2002 in Grenada, and ever since then has been spreading west and north, finally being reported here in 2012 in St. Thomas. A really fun fact that I thought was neat is that it invaded the Mediterranean over 100 years ago through the Suez Canal. So we're not the only ones in this journey. Halophila stipulaceae has a lot of unique traits that kind of set it aside from the native seagrasses like Thalassia, Serengodium, and Halophila. One of these really cool facts is that it's been reported to grow deeper than 50 meters. Unlike the really lengthy native seagrasses that we have, these guys are small and they're also dioecious angiosperms. That means they have both male and female flowers. Similar to the native seagrass Halophila decipiens, Halophila stipulaceae is shallow rooted. This means that it can be easily uprooted and fragmented. And what that means is, and while that might sound like a bad thing to us, it's actually not terrible for them. What they do is after fragmenting, they'll float, root, and colonize in a new area. This supports the theory that around St. Thomas, Halophila stipulaceae populations have low genetic diversity due to their primarily reproducing asexually. Additionally, they have fast recovery rates following small-scale disturbances like fragmentation. However, Kyle Jarris in 2019 released his thesis where he suggested that after the 2017 hurricanes Irma and Maria, Halophila stipulaceae bounced back faster than the native seagrasses. We're concerned about Halophila stipulaceae for a few different reasons. As you may know, they are considered invasive seagrasses, and that means that they cause problems in the natural environment. For example, Halophila stipulaceae competes for space with native seagrasses, causes shifts in seagrass, fish, and invertebrate community composition, and we've also noticed that they like to avoid certain herbivores, like to avoid eating Halophila stipulaceae. This could be because they have what's considered low palatability. In fact, in a study by Christianen in 2012, they found that Halophila stipulaceae would rather eat native seagrasses like Thalassia and Serengodium, green sea turtles, than Halophila stipulaceae. Herbivores are really important for shaping the structure of a seagrass meadow because they graze on the seagrass. The idea that an invader becomes more successful when grazers aren't offering that top-down control is the idea of the enemy release hypothesis. So I use this term palatability, and I'm sure you're just sitting there going, what does that even mean? So I'm going to explain it to you, and I'm going to break it down. Palatability is the word that we use to describe the susceptibility of a plant to being eaten. It's a little hard to define because it's not only relative to the consumer, it's also relative to the plant. Consequently, this means that when you're studying palatability, you can't just look at one consumer. Just like humans, animals are picky. One animal isn't going to like the same thing that another animal likes. So you have to look at them under the influence of multiple consumers. However, in general, it can be defined as a multifactorial feature of an organism under attack, resulting from chemical and physical characteristics of a tissue, or how yummy is the plant to the consumer. So a lot of what we know about top-down control has been studied looking at plants of different species. For example, let's say we have a seagrass meadow and we have species A, and the herbivores love eating species A, but they don't really like eating species B. We could come to the conclusion that species A is more palatable than species B. The same thing can be seen in plants of the same species. Nutritional differences, light intensity, and even salinity differences can make a plant of the same species taste different to a different consumer. This is the idea, and I know you can't really see it, I apologize, of intraspecific variations in palatability. That's going to be the main focus of this thesis. Unfortunately, though, the enemy release hypothesis has only focused on interspecific differences, so those between species, even though intraspecific differences in palatability can be just as high. I'll take a sip of water real quick. With all of that in mind, I wanted to try and explain the intraspecific palatability of Halophila stipulaceae as it relates to the enemy release hypothesis. So using these objectives, first I wanted to look at quantifying intraspecific variation in palatability of the invasive seagrass, and then I wanted to relate this variation in palatability to the enemy release hypothesis. These objectives can be reflected in my experimental hypotheses. So number one, palatability of Halophila stipulaceae will vary with depths within sites. So this is taking one day and looking and comparing all of the different depth ranges that I use. Two, palatability of Halophila stipulaceae will vary when plants growing at similar depths, but from different sites are compared. So this is taking multiple sites and comparing them at similar depths. These first two hypotheses were done in laboratory settings. However, I also wanted to look in the field. So differences in palatability among depth and sites will be reflected by grazing patterns in the field. Okay. You guys all know before we start, we got to figure out where we're going. So while we're going on this journey, we went to Brewers Bay, Lindbergh Bay, Frenchman Bay, and Water Bay. These sites were chosen not only because of their diverse range around the island, but also because of their accessibility and the fact that I knew that there was a Halophila there. I also chose 3 depths. Again, I apologize, you guys can't see this. Blue is shallow, so less than or equal to 3 meters. Green is medium, so 8 to 12 meters. And pink is deep, 15 or greater than or equal to 15 meters. In Frenchman Bay, I only collected from shallow and medium. That's because Halophila and the bay itself is not very deep, and so we didn't include that category. That's important to keep on to. Obviously, if you're testing palatability, you've got to have some hungry animals. So I chose 3 crabs based off of their abundance around the island. They're known, they interact knowingly already with Halophila stipulaceae, and because they're really good lab animals. So first we have the adorable Homolacanther bicornata, or the speckled clawed decorator crab, the grumpy Mithraculus sculptus, or the emerald clinging crab, and the prickly Echinometra loop hunter. You guys probably know these as the rock-borne sea urchins if you've ever stepped on them. I have. So these animals were collected via hand from Bruce Bay and Black Point Bay. Seagrass was collected snorkeling and via scuba diving. I didn't do the scuba diving. I have some of these lovely people in the audience who did my diving for me, and we collected them haphazardly. So what that means is either myself free diving or one of my lovely divers would go down, grab a handful of seagrass, put it in the bag, swim about 10 paces, grab another handful of seagrass, put it in the bag. And this allowed us to not only look at one spot on the meadow, but the entire meadow at that depth range. Now, there's a lot of different ways you can measure palatability in a lab setting. I decided to do what's called paired choice experiments. This is because I wanted to look at the pairwise differences of bays and depths. These trials are typically run for 48 hours, and they are set up like this. So you have two water tables, or I use two water tables, and in each water table, there were 40 plastic tubs. That equates to 20 replicates. Each tub had its own water flow. Before the experiment started, seagrass was weighed and added to the chambers. They were held down by Velcro because, as we know, Halophila floats. After that, we added the animals, started the timer, and 48 hours later, I would come take down the chamber, take down the trial, and weigh the seagrass again. So something that we have to consider when running these is that we have to account for the autogenic changes in seagrass mass. To do this, we would add controls to each replicate, and these controls had no grazers. So we were allowing them to do what they would naturally do if there were no grazers present. Once we had all of that, I calculated amount consumed. And since I was giving the animals two choices, I was able to run paired t-test. The fun part. Okay, so the first few graphs are going to be showing you within bay depth comparisons. So for these, I ran a total of 3 different permutations: shallow to deep, medium to deep, and deep to shallow. Now, when you take those 3 permutations, add the 4 bays, and then the 3 invertebrates, that's a total of 30 experiments. On the x-axis, you guys will find Bruce Bay medium, Bruce Bay shallow, Bruce Bay deep, Bruce Bay shallow, and Bruce Bay medium, and Bruce Bay deep. The x-axis will always indicate what trials were being run. The y-axis is showing Halophila consumption, which is measured in milligrams per animal per day. The top blue bar is the sea urchins, the green is the clinging crabs, and the bottom are the decorator crabs. Now, spoiler alert, most of my experiments look just like this. No significance, right? Nothing's going on. There was no preference made between the 2 options given to the animals. But there were a few interesting bays, and that's what I'm going to share with you now. Lindbergh Bay was one of those bays. So on the y-axis, you'll find the trials medium to shallow, shallow to deep, and medium to deep. The urchins were not selected in this experiment. However, the emerald crab did show preference, significant preference for Lindbergh Bay deep over Lindbergh Bay shallow, and Lindbergh Bay deep over Lindbergh Bay medium. Additionally, the decorator crab also showed similar preference, choosing Lindbergh Bay deep over Lindbergh Bay shallow. Then I also saw something, and I should mention this. Remember, I only took shallow and medium for Birchman. So those are the only within bay comparisons that were made for this bay. But I did find significance where the emerald crab preferred Frenchman medium over Frenchman shallow. So for within bay comparisons, as you could tell, most comparisons did not show any preference or demonstrate any differences in palatability. However, there were 4 out of 30 experiments that did stand out and show significant preference by the 2 crabs. And 3 of those were Lindbergh Bay deep. Hold on to that thought. Lindbergh Bay deep, just let it soak in. For Brewers Bay comparison, so not Brewers Bay, sorry, between bay comparisons, right? So I also did those. This is where, again, we're looking at all the different bays and comparing them at similar depths. So I never crossed the depths. I never did like shallow and deep or medium and deep. We just wanted to see if they were same across the board. So on the bottom axis, you're going to find these trials as water medium, Frenchman medium, water medium, Brewer's medium, Lindbergh medium. That's what they'll look like. Same thing on the y-axis. We have halophila consumption. The animals are still the same color. I will go ahead and point out for the between bays, because there's more comparisons, there were 45 experiments run. And spoiler alert again, most of them look just like this. However, drum roll please, because some of them didn't. So we're going to start by looking first at the decorator crab. For water bay to Lindbergh bay, they chose water bay medium. And they also showed significant preference for Lindbergh bay over Brewer's bay deep. The emerald crabs this time weren't that hungry, I guess. They didn't show any preference for the two options given. However, the sea urchins did come out to play, and they chose Lindbergh bay deep over Brewer's deep, Lindbergh bay deep over water deep, and they also showed a trend suggesting that the urchins do have a tendency to also prefer Lindbergh shallow over Frenchman shallow. However, that's not significant. So again, most comparisons for the between bay trials did not demonstrate differences in palatability for Halophila stipulaceae. However, 4 out of the 45 experiments did. And again, 3 of those 4 were Lindbergh bay. The only difference here was that the urchins showed preference, whereas the emerald crabs didn't. So Lindbergh bay deep, let's talk about it. Lindbergh bay deep was preferred by all 3 invertebrates during some point within and between bay trials. I cannot for sure tell you why that is yet. However, there are some key features of Lindbergh bay that could help us explain and shed some light on why this is. Lindbergh bay is located on the southwest side of the island and is surrounded by the airport, a really popular beach, a really busy road, 2 major hotels, a really annoying jet ski rental, if you know, you know, and a water desalination plant. So we do know that the airport waste does flow into this bay, and one ecological consequence of a water desalination plant is poor water quality from the areas it takes water from. DPNR in 2010 released a report suggesting that turbidity and recreational pollutions are considered water quality impairments for Lindbergh bay. And it turns out, as light decreases with depth, nitrogen in some seagrass species increases. This is because deeper seagrass, some species of deeper seagrass, have more chlorophyll A to enhance photosynthetic abilities. This increase in nutrients could explain why Lindbergh deep water or Lindbergh deep seagrass was preferred by all 3 invertebrates. Another sip of water. Speaking in a mask is not easy. Well, all the teachers out there. So thus far, we've looked at experimental data that I've collected through experimental trials. However, that's only half of the equation. While the experimental data explains to us the feeding behavior of certain organisms, it doesn't show us what herbivory pressure is like out in the field. So to do this, to bridge this gap, I decided to go back to the same spots with my divers and myself and collect again from the same exact way. On the same day of collection, I scanned the seagrasses onto a Canon Scan 300 as shown in this beautiful picture of me. I then processed them with image J. On image J, I was able to look at numerous different measurements, and some of those measurements were components of herbivory damage or herbivore damage. So I quantified percent of leaves with grazing scars, mean number of scars per leaf, and I was also able to look at the approximate area of missing leaf or area removed due to herbivory. So I told you to hold on to this. Frenchman Bay, I said it a few times now, but remember I only collected from shallow and medium, not deep. This becomes really tricky when you're doing multifactorial ANOVAs because they're really sensitive to missing data. So before running the 2A ANOVAs, I applied a protocol called MMES, or multivariate imputations by chain equations. Multivariate imputations by chain equations. It's a mouthful. That's why we call it mice. So mice was applied to account for this missing data, and then two-way ANOVAs were run. So all of the data you're going to see from now on has this protocol applied. On image J, one of the areas I got to measure that was really interesting was area of missing leaf due to predation. This blue arrow is pointing to what I consider a lateral scar or a measurable bite mark. The pink arrow is pointing to an apical scar or a non-measurable bite mark. So what's that saying is I didn't feel comfortable assuming leaf length when the bite was off the top. So whenever I talk about area of missing leaf, I'm only talking about lateral scars. And I'll remind you, I'll bring it back up. So again, getting into the fun stuff, I looked at percent leaves with grazing scars. Now I took this by taking the ratio of total leaves with bite marks to the total number of leaves. There was a significant interaction between bay and depth. However, when I ran the post hoc test, it wasn't able to pick up on these differences. That's pretty usual though, since the statistical power of a post hoc test isn't as strong as an ANOVA. If you do look at the graph though, you'll see that on average, Frenchman Bay and Brewers Bay had more leaves with grazing scars in the shallow depths, and Water Bay had more leaves with grazing scars in the deep. So that's the opposite to Brewers and Frenchman. I took all of the samples and I wanted to calculate total percent leaves with bite marks, and I got 11.53%. Remember that because I'm going to explain why that's crazy later. So in addition to percent, I wanted to look at mean number of scars per leaf. I had no, and oh, I should mention that the x-axis is bay, Brewers, Frenchman, Lindbergh, and water, and pink is deep depths, green is medium, blue is shallow. I apologize if you weren't able to follow along. And then the y-axis here is the mean number of scars per leaf. So that's what we're looking at here. There was a significant effect of bay. However, there was no significant effect of depth, and there was no interaction between the two. On average, Frenchman had more scars per leaf than Brewers Bay. Lindbergh Bay and Water Bay are just hanging in there with intermediate number of scarring, and they were statistically equivalent to the other bays. Finally, I measured approximate area removed. So if you look up at this little picture, it's just reminding you that I only calculated lateral scars. This is important because Brewers Bay Deep is missing from this graph. So Brewers Bay Deep did have scarring. However, it didn't have lateral scars. It only had apical scars. So it's not included in this graphic. Mice was applied. And so now it looks a little silly. On the x-axis, we have mean area of missing leaf, which is calculated or measured in centimeters squared. There was no significant effect of bay or depth, and there was not a significant interaction between bay and depth. Nothing significant. However, if you do look at the graph, you'll see that Brewers Bay did have more variance in the number or area of bite marks removed. So I told you to remember the 11.53. There will be a quiz at the end. This is an important number because I calculated that 11.53% of the samples collected around St. Thomas had leaves with scarring. In a study by Mariana Alcavero in 1989, they found that there was up to 50% leaves with damage. This is interesting because this study is done in the native range. So just an interesting comparison because 11.53 is really low compared to its native range. Overall, grazing was low in the field, which supports the enemy release hypothesis. But in order to really confidently say that, we do need to do similar research on native seagrass species. Contrary to what I thought was going to happen, Lindbergh Bay in the field didn't show exactly the same things in my experimental data, where everybody loved it. Here it's just, eh. So it showed similar grazing pressures to that of other sites and depths. This could be for a number of reasons. One of those could be that there's just not that many herbivores in Lindbergh Bay, or that the distribution of herbivores is different than those of Brewers, Water, and Lindbergh, or Frenchman. In conclusion, plants from different depths and bays generally did not vary in palatability. This agrees with the general idea of low genetic diversity within these populations due to asexual reproduction, like fragmenting or budding. Lindbergh Bay meadows appear to experience environmental conditions that enhance palatability. This is only for laboratory experiments, because like I said, for the field data, Lindbergh Bay didn't show any differences. Love that construction noise in my dreams. Field patterns of grazing pressure showed little variability. And the enemy release hypothesis does not appear to be offset by population or genetic differences in Halophila stipulaceae palatability. Now, I really like the idea of discovering what these trends in Lindbergh Bay actually mean, but also looking at palatability on a larger scale, such as looking at the population here and comparing it to populations on other islands, or using different grazers. But to do that, we need to understand palatability in a different way. We need to understand the multiple factors that contribute to palatability. There's a few different ones, and I'm going to talk about them right now. So morphology, or the structural differences of a plant. This could be anything from what it looks like to what it feels like. So toughness is often one of these. If something's really tough, you don't want to eat it. The same thing goes for seagrass. Nutrient content. We kind of touched on this a little in Lindbergh Bay, where we talked about some plants have a higher nitrogen content. So nutrient content is a known cause for palatability. Opposite to humans, we don't always eat nutritious food. Animals have to. And then we have chemistry. So some plants have chemical defenses that will deter herbivores. And even genotype can be used as a proxy for palatability. Something that I'm really interested in doing with my samples is looking at bite mark identification. So trying to determine who is eating my seagrass. This is hard to do because sometimes bites look really similar. And so I don't like to just look at a seagrass and be like, ah, sea turtle right there, you know? So, but Prado et al. In 2007 actually was able to do this with their seagrass. So they were used different identifiable markers and were able to tell that A is a juvenile fish bite mark and B is a sea urchin bite mark. So it'll be really interesting to go out into the field, catalog which herbivores are there, come back, look at the data, and determine who's actually eating the seagrass and offering top-down support. So if you guys were here earlier in the summer, you know that the -80 freezer broke and I lost all of my nutrient samples. I would have loved to share these results with you, but I'm just not working on them. So I am profiling the nutrient content of Halophila stipulaceae, and I'm also working with UC Davis to run genetic analysis to determine if there's any differences in palatability that can be detected by genotype. Ultimately, though, other potential areas of work that need to be done are field tests on the enemy release hypothesis. This will help us explain the magnitude and pros and cons and impacts of this invasion of Halophila stipulaceae. So with that, I want to thank first the money. That's important. So thank you. I know you can't see the logos, but we've got EPSCOR, the Lana Vento Charitable Fund, and the NSF STEM grant. I also want to thank a special shout out to the Polk Education Foundation for not only funding my master's degree, but also my bachelor's degree. Thank you to my advisor for everything. My committee members, Dr. Stanford and Dr. Turner, for helping me with my writing and my experimental design and just keeping me calm, which you know is hard. Also, thank you to all these people listed. I really do want to shout out Sophie Costa, Kelsey Vaughn, Alexis Long, I'm assuming she's there, Kayla, and Stephanie Hibberts. Not only were you guys my rocks, but you guys also worked your field schedules around my fieldwork. That's really helpful. Thank you to all the people who helped me, Kaya, Chris, Amika Parr, who was my undergraduate assistant for like a month. Also, thanks Ian, Amber, and Adam for all the help with boats and water table flow, fan of my existence. Thank you, Joe and Dr. Lombardi, for helping me with my statistics. And thank you to my family for just being there. It's been a rough 2 years. Awesome. Well, I will now take any questions.
It must be so hard to be hauled around all these places like Hawaii and.
I know.
Oh, okay.
Wow.
Yes.
That's not the hard part.
Any questions? We really need, Sophia, are you monitoring the people online to see if anyone?
I can
has
do it. They can
questions?
also just speak. We can hear them.
Yeah, I can help do that too. So far I'm seeing lots of great job. You did awesome. Fantastic introduction to Edwin. That was a good laugh.
Also, good luck to Edwin. I love the username. Thanks, Jen.
All right, questions. We have time.
You have a question?
Yes.
Okay. I'm nervous.
I'm just curious. Do you know, you might not know, maybe they didn't mention it, but in that study, it was MNA something 1999.
Okay, yeah.
How did they measure like bite marks in the same way? Is he like not measuring the topical or the apical?
That's a really good
There we
question.
go. Or because like
I think that
maybe
they
that's why
were.
yours is so much lower, or do you know if they did it differently and like included that somehow?
Yeah, so with the percent and the mean number, that's including all leaves. Yeah, so the only time that I took out the apical scars was during when I was measuring area.
Okay,
Yeah, so
gotcha.
they were included in the percent and the mean. Yeah.
Okay.
I would assume that the study in 1999 also looked at all scars, not just certain ones. I don't remember off the top of my head though exactly how they counted.
I was just curious, but not
Yeah.
clears it up. Thank you.
You're-
I wasn't sure if you used all of
I
it.
should have mentioned that. Thank you.
Was there a difference in the amount of epiphytes in the seagrasses from one location to another? And do you think that might have anything to do with differences in feeding?
It's a good question.
Because of like a lot of invertebrates might graze on the epiphytes and then graze on the seagrass
Indirectly, yeah.
indirectly as a consequence of that.
Yeah, so I definitely think that's possible. I'll say, based on observations, Lindbergh Bay has lots of epiphytes, especially in the shallow and medium range. So does Water Bay. So I definitely think it's possible that epiphytes play a key role in grazing. It might not be that they're eating the leaves, they could just be eating the algae that's stuck on the leaves. So yes, I do think it's possible. I didn't look at that in my study. However, I did think about it often when I was like cleaning the epiphytes off my seagrass. I was like, I wonder if they're eating this.
So you clean the epiphytes?
I did. I
off of the
cleaned
seagrass
seagrass.
producing them?
Yep.
Okay.
So they weren't given, at least to the best of my ability, they weren't given algae covered seagrass or epiphyte covered seagrass. Yeah,
Gotcha.
but I definitely think that could play a role. And I also think nutrient content could play a role in the like abundance of epiphytes. I'm sorry. Your eyes. What?
What was your feeding schedule for invertebrates? And did you ever deprive them from food for a certain period of time before you fed them? Did they starve?
I did not starve them. Did they starve? No, I fed them regularly, probably I would say weekly. They got about 2 different species of algae that I collected from the intertidal zone. I never fed them other seagrass though, only algae. They're really fast eaters, so you do have to keep the tank rather full. I didn't starve the animals before the experiments because there's a lot of research that suggests that that can create an unnecessary bias. So I decided to, also in their natural environment, they don't starve themselves. So I wanted to keep it as natural to how it would look in the field as possible. So no starving happened. They're all really happy. If you go to the Marine Center, you'll see that. They stand on the rocks and like wave their arms around for the food. It's really cute.
I just have a slight question. So your Frenchman bite marks, right? That had the greatest area.
Mhm.
Okay.
Or the
So-
wait.
When you're talking about comparing the different species, so I don't know, because I'm doing those food traps.
Are you
I
looking
don't
at this
know
one?
If the species, well, just the bite marks in general.
Okay.
And then didn't you say like your
Yeah,
Frenchman
Frenchman
area?
has the most scars. Yeah.
Okay, so like in my food traps, I wonder if some of the like individual species you're looking at, I don't know if these are like large inverts or Like, are you looking at just like any sort of like snails? Like what has
It
a
could be
preference to those?
anything.
I don't know anything about the difference.
Yeah, so I kind of mentioned this. So at the beginning, I mentioned that when you're studying palatability, you have to look at more than one consumer because everybody's different, just like people. So I study these 3 invertebrates. While they're grazing on it, it doesn't technically mean that if they're in the field and they see a Thallassia over Halophila or whatever, that they're going to eat Halophila, because why would they if they don't want it? But I would say that part of me identifying bite marks could be looking at snails and other animals that are known consumers of Halophila, but also larger animals, because sea turtles also eat the Halophila. I mean, I've seen it. Actually, it was in your video. There was a turtle eating Halophila, and I was like, wow, look at that. So yeah, I can't tell you for sure.
Yeah. It's just what's really interesting to me about your results is that as far as species, like fish species richness, Frenchman's has such higher fish species richness than Lindbergh.
That makes sense then why I'm seeing this.
Yeah,
because I definitely, fish are a player that I think, I forget about a lot because I don't study fish. I'm always like invertebrates and sea turtles, but yeah, no, fish. And I didn't even think about that because Frenchman Bay also has that shallow reef.
Yeah.
So it definitely could be that it would be different results if we looked at fish.
I think that
Yeah.
we have a paper.
I'll fly back for you.
Yeah.
Are the fish actually actively eating a seagrass or like the epiphytes on?
Great question. I don't know.
It
It could be both, because Frenchman Bay, they have epiphytes, but it's not, I wouldn't say it's as bad as as Lindbergh Bay, where it's just, it was hard for me to find patches where it wasn't epiphytes. But yeah, I mean, they definitely could be eating the algae, especially parrotfish maybe.
Yeah, I've definitely seen like Judy parrotfish like munching on the Halophila in the field. So
That's good.
Yeah.
I'd would like that because I know you want to look at parrotfish. It's cool. I don't know.
Any other questions?
No, just Sophia putting in the survey. Everybody go take your survey.
I have a question.
Hi.
Awesome.
Hi. So I'm not sure if you were in the literature review presentations this morning, but the 2021ers started their lit review presentations, and one of them was talking, to be honest, it's gonna take me a minute to think of who it was, but one of them, I think it was, I believe it was Taylor, was talking about differences in seagrass nutritional value. And specifically, the thing that was kind of interesting about it had to do with how the younger plants, the shoots, have a higher nutritional value than the older ones. So, and I think they're talking, I think that paper was talking about sort of nutritional value within the leaves, not considering the epizoic, growth on it. But I'm just wondering if you, by any chance, I know it's off topic, but by any chance, if you looked into anything about the nutritional value of Halophila compared to the other species of seagrasses.
Yeah, that's a great question. So I am currently processing nutrient samples. I'm specifically targeting proteins, carbohydrates, and lipids. It is thought that certain herbivores will specifically eat things that are higher in protein, for example. So I will say there are some studies that have looked at Halophila, stipulacea, nitrogen, and phosphorus content compared to like Thallassia and Ceragodium, and it's always rather low. And it matches the whole preference where they choose other seagrasses over Halophila stipulaceae. So I think that's interesting about the young and old. I didn't ever think about that in plants.
Well, we talked about it, but you don't have the data. I do.
Oh, okay. I'm Edwin and I have all the data. So yeah,
right, Madyson. One of the things that got me about her discussion was that when I think about how these different species grow, I get a completely different, sense of the morphology of a shoot of Thallassia and how it's different than the mature leaf compared to Halophila. Because, for example, where is the shoot?
Oh, I can
It's-
show you.
So, in other words, but it doesn't seem that dramatically different between the shoot and the mature leaf, right?
Yeah, so if I'm being honest, Lori, I don't know too much about the nutritional value of the actual shoot themselves. I am looking at the actual nutritional value of the leaves. But
Well, I mean, the leaves are part of the shoot, but I guess she's talking about I guess she's talking about that early, like in that particular photograph you've got up now. I think there's one leaf there that's very young, right?
yeah, like these
On the
little right
left, yeah, exactly. But we don't really see a new shoot forming. But
Mhm.
when you see a new shoot forming, it would look like that only a single,
itty bitty.
only a
Yeah.
single blade there, right, right. So, but then when you think about the difference in the Thalassia, I mean, sometimes there's a couple of orders of magnitude difference in size, and then there's the difference in how the old Thalassia blades get kind of gnarly and I would think are dead
Yeah.
at the tips.
So
So
morphology definitely plays some sort of role, maybe not alone, but in the presence of nutrients and genetic variations, morphology is a contributor to palatability. So it's likely
Yeah.
that, yeah. I
So
did take
it might affect them. I don't know. The other question I'd have about palatability is about do you think that the amount of growth of other species, other maybe algal species or animal growth like bryozoans or something on these seagrasses makes a difference in palatability?
I definitely think so.
Okay.
Yeah, I think so.
Is that a gut feeling or you have some evidence?
I think that's a gut feeling. I can't actually back that up right now. Maybe if I went through my source of papers.
Well, the
But
gut feeling is important, Madyson, because, I guess it's the Germans actually named it Gestalt. It's it's an important feeling. It might be something to follow up on for your next project. I don't know. But
I will
a
follow
very
my
interesting
gut
presentation, and I'm sorry to take you off topic, but it just
No,
came to
thank
mind
you.
when you were talking, and I would encourage you to sort of get in touch with Taylor. Last name.
Yeah, I'll get in touch
either
with you.
Brunson or Hobbs. I think it's Taylor Brunson that went this
Oh,
morning. I'm
not Taylor.
not
Got it.
great with their names yet, but I'll link you two by email. Okay.
Okay, awesome.
So-
Thank you. That was great.
Oh, Edwin wants to say something, Lori.
So we
Hi,
have those
Edwin.
data from a couple of summers ago.
What's that?
We have those data for a couple of summers ago for.
Oh, you do?
their
Okay.
younger leaves.
I'm gonna loop you into that email then.
Yeah, but it was basically the same grazers that Madyson was working on. At one point, we added, we thought about adding that comparison to her thesis, but I think that 75 experiments was enough for her.
I think so too. I
7 months
absolutely
ago,
agree. Thank you for that, Edwin. That's great.
Awesome. I think. Any more questions?
Great job.
Great job, Maddie.
Thank you.
Any other questions
I
online?
forgot.
Sophia, we cannot monitor that from here. Can you
I can.
check whether?
I don't see any more questions in the chat. I did put some survey links in the chat, and I will go ahead and send by email to those of you who are in the room. Lots of congrats.
Oh, look, Jordan's here. Hey, Jordan.
Awesome. Well, thank you very much to those of you who came in person and those of you who attended the fence online.